I'm going to introduce nanobodies. In the biomedical literature, this is a sentence that you read quite often. And it deals with proteins within cells. This is a sentence that you read quite often. These and other findings suggest that inhibitors of protein X, Y, or Z may offer an effective way to target some malignancies. And so to date there is an avalanche of manuscripts claiming that a certain protein is an interesting therapeutic target. So what we try to do in my research group is to develop tools to target proteins that are difficult to drug. And by that I mean structural proteins, not enzymatic proteins, because most pharmacological inhibitors, of course, they target enzymes. And we apply this technology in the field of cancer, but also inflammation, and I will give you one example of an amyloid disease as well, similar to Alzheimer's disease. The idea is that we try to block functions of proteins, thereby triggering what is known as a protein domain knockout, not using pharmacological compounds. And so what we try to do is to use so-called nanobodies or single domain antibodies to achieve this goal. What is a nanobody? Well, basically, it's a fragment of an antibody. And camelids like alpacas, vicuñas, and camels and dromedaries, they have two different types of antibodies. One of them, the first one, is a typical conventional antibody consisting of two heavy chains and two light chains, but in addition to that, they also have what is known as a heavy chain only antibody. And this is an antibody that lacks light chains. And it is possible to clone the antigen binding part of such a heavy chain antibody, and that is known as a VHH or a nanobody. What is very important is that it is easy to clone this fragment. You can obtain the cDNA. They're very small, 10 times smaller than a conventional antibody, only 15 kilodaltons in size. They're easy to produce in high quantities, relatively speaking, of course, 10 to 50 milligrams per liter of medium in bacteria. They're very stable, specific, and it is possible to affect functions of proteins. It is possible to affect functions of proteins, non-enzymatic proteins. And this is a Coomassie-stained gel showing the purification or a purified nanobody from bacteria. Incidentally, 5 years ago, two American researchers, Gobilka and Lefkowitz, they received the Nobel Prize in Chemistry in 2012. The reason is very simple. They were the first to determine the X-ray crystal structure of a GPCR. And that's a G-protein coupled receptor. And those receptors are very important therapeutic targets. It's very difficult to determine the X-ray structure of a GPCR because it has many different conformations. And so they use the nanobody as a G-protein mimetic in order to keep the GPCR still, quiet, so that they were able to determine the X-ray crystal structure. But this is just one of the many applications of a nanobody. Planetary scientists, they use rovers to scan the surface of a planet. And so what we basically do is the same, but we use a nanobody to scan the surface of a protein and to find an interesting therapeutic epitope. But basically it's the same. So you can use a nanobody as a molecular charting device to identify functionally important regions in a protein. I'll just give one or two examples to illustrate this. Here you can see a control group of mice, and they were injected with human breast cancer cells. So these breast cancer cells will grow as a tumor, and over time you can see the size of the tumor increasing. Eventually the animals will die because of metastasis. Metastasis, or the spread of cancer cells, is the most important cause of death in cancer. And you can see this here. The two lobes represent the lungs. So the tumor cells escaped from the primary tumor and they migrated to the lungs. And because of this, the animals will die. Now, the upper panel is a control group, and the lower panel is a group of mice that were treated with a certain nanobody that affects the migration or the motility of cancer cells. And you can see that in this group, there is no metastasis whatsoever. So the cancer cells, they do grow, but they do not migrate to other regions in the animal. What we do next is we determine the X-ray crystal structure of the antigen in combination with the nanobody, and this identifies an interface between the nanobody on the right and the antigen on the left, and this shows you a therapeutic epitope, because the binding of a nanobody to this region of the antigen translates into a very interesting therapeutic effect. This is just one application. We apply this in the field of cancer cell metastasis, but also cancer cell exosomes, biogenesis and release of exosomes from cancer cells, in addition to selected signal transduction pathways, including P53, AKT, and survivin. This is an entirely different research line where a nanobody is used as a diagnostic. As a diagnostic in the field of amyloid diseases, and the Alzheimer's disease is one of the many examples of a typical amyloid disease. And so here we have a mouse model for a particular amyloid disease, and we tried to use a nanobody to identify the regions in the animal where these amyloid deposits are present. And this can be done using a nanobody that is labeled with a metastable variant of technetium-99. In combination with SPECT and CT imaging, you can see these blue-purple regions in skeletal muscle, and this is exactly where the amyloid deposits are present. So, in principle, this can also be used for other amyloid diseases, such as the disease of Alzheimer's, to identify in individual patients where the amyloid deposits are located. I'm not going to discuss this in detail. It just gives a very recent overview of several ongoing clinical trials where these Camelid nanobodies are used for a range of different diseases, including rheumatoid arthritis, psoriasis, which is a kind of skin disease, asthma, bone disorders, different types of cancer. So maybe in the future, these molecules may develop not only in diagnostic molecules, but also in therapeutic molecules as well. So to summarize, I tried to show that nanobodies, well, I didn't explain this one, but it's not too important. A nanobody can be interesting to predict drug effects, and it can also be used to predict side effects of drugs. It can be used as a therapeutic, possibly in the future. It can assist in compound design, medicinal chemistry, and they are very useful in co-crystallization experiments because many proteins are very difficult to crystallize. And if you add a nanobody against that particular protein, it may be very conducive in obtaining the X-ray crystal structure of a GPCR or another protein. I haven't discussed this in detail, but they might also be very useful in the future in what is known as super resolution microscopy. Okay, I think I'll stop here. And I'd like to acknowledge these people, particularly Wouter and Adriaan, who were responsible for most of the work and different collaborations in Singapore and Canada. And I thank you for your attention.